where can I, oh, there we go. All right, so is it full screen now?
Yeah, it looks good.
Okay, cool. All right, so, just as a brief recap, genome-wide association studies are, have become probably the most common study design to assess any link between genetic variation and complex disease. And the way they typically work is you assay genetic variants in a large number of individuals, in a case control cohort, and you simply march along the genome, scanning, performing correlations to see if anything pops up at a very strict significance threshold. This works really well. It relies on really simple models, and things tend to be somewhat robust, or more than somewhat. I'd say things tend to be robust and replicate well. But the primary challenge with GWAS is that it really provides regions associated with disease risk. Just because an individual variant has statistical support for an association doesn't really mean that it's causal or actually driving the risk at that region, or explaining the risk at that region. And the reason is largely due to LD, right? Inheritance patterns are that we inherit genetic variants from our parents in chunks. And because of that, they are correlated in the population. Where these sort of chunks are determined is largely a function of complex biochemical processes, natural selection. And so these regions tend to be similarly inherited across individuals in a population. So what we see are these population level correlations between genetic markers. And so given that, what we'd like to do really from GWAS is try to understand what the mechanisms driving disease risk are, right? Most associations are non-coding. That means they don't change the chemical composition of the coded protein. And there's plenty of evidence at this point that the SNPs are regulatory in nature. And what I mean by that is, rather than changing the composition of the protein, they regulate the expression of some gene to some degree. So you can think of it upregulates or downregulates the amount of some mRNA that gets produced and eventually translated. And so here, we see a screen cap from some schizophrenia GWAS for a few years back on the difficulty of just trying to figure out what gene is relevant at a risk region. It's not as simple as picking the closest gene. That might be a really good heuristic that works well in practice, but there are certainly plenty of instances where that will fail. And just, as a nice illustration, right, if we just try to eyeball it, right, looking at this risk region here, each point is a genetic variant. Its height along here is evidence of association. And down here are all the genes at this locus, right? And so if we were just to try to pick which gene is explaining risk, we really are sort of just out of luck without any sort of additional functional support. And so one way to get around this is rather than testing genetic variants, we can just test gene expression levels, right? If it's the levels of expression that correlate with disease risk, why not just test that? Well, the problem is oftentimes we don't have that, and measuring it's very expensive to get it at scale of GWAS. And so one way to get around that would just be to predict it using genetic variants. And so this is, several papers proposed a few years back where you go into smaller reference panels where you have genetic variation, you have expression of some gene, you fit some penalized model, some predictor under a linear model, you get your weights. So here beta are, we can think of the effects of each genetic variant on the expression levels, and we go back to our individual GWAS data, right? Our case control cohort, we impute the expression using their genotype, these weights, and now with this imputed expression, we simply see if that tracks with disease risk. Right, and so this kind of mode of thinking, or this design, is now probably mostly referred to as a TWAS, or a transcriptome-wide association study, or a TWAS using predicted expression. And sort of the nice thing about this is that it actually isn't reliant on requiring individual level genotype data in your GWAS cohort. Due to the linearity of these predictors, you can actually just take summary statistics that are published from your GWAS, right? And so if we go fit our predictors, we download the z-scores for each SNP. If we just apply a linear transformation of the z-scores under some certain assumptions, that ends up being equivalent to just predicting the gene expression in the original cohort and like projecting that onto the phenotype, right, to get some sense of an effect. The issue here is that LD needs to be taken care of, but this can be dealt with through external reference panels like 1,000 genomes. Right, and so the nice thing about this is now you can just build your predictive database, download some summary statistics, and apply it, right? And so one thing we did several years back was apply this to prostate cancer, and we identified several risk regions. And so here, what I'm showing is a bunch of genes at this locus. Each point is the genome-wide association signal for a given SNP. In gray was the original signal, and then in green is if we regress out the predicted expression, right? So you can think of conditioned on the predicted expression, what happens to the residual GWAS signal, it's largely null. And so, this is consistent with expression of MLPH, increasing risk for prostate cancer. So that's all great. That's kind of the background for TWAS. And I did give some background earlier on why we see correlations at risk regions due to LD. And so what I'm showing here is commonly referred to as a Manhattan plot. Each point corresponds to a SNP. We see these towers that look like buildings, hence the Manhattan name, because of these LD patterns, right? And so if I were to show you this, and everyone say, oh, this is just your typical GWAS Manhattan plot, but in actuality, what happens is this is a TWAS Manhattan plot. Each point corresponds to the association between predicted expression, and below it is the corresponding GWAS SNP-based association. And so you see these similar patterns at where associations are correlated at risk regions, right? And so we saw this earlier on. And I think, some of the surprise for this in the general community was that TWAS is supposed to give causal associations when that's not clearly the case. It's right there in the name. It says association study, not causal gene identification study. And so what we wanted to do is come up with a method to sort of figure out what's going on, right? To given these associations in a TWAS, can we provide evidence of what the causal gene actually is, right? And so here's just kind of toy cartoon that could explain this a bit clearer. Each point, again, is a SNP. Its height on here is its strength of evidence for association with the trait. Down here, we have our weight expression matrix, where we have 6 genes. Gene 3 is the causal gene at this locus. The darker colors indicate some weight that a genetic variant regulates this gene. And down here, we have the correlation matrix, right? That's showing the LD, right? The correlation between the genetic variants. And so if we were to perform a TWAS one at a time for each of these genes, what happens is we see these associations, right? So we do see that our causal gene pops up, which is great, right? So TWAS is sensitive, but it's not specific, right? We have these other associations also popping up. But it turns out, because again, we're taking linear transformations, we can actually pretty easily figure out what the predicted expression correlation matrix should be, right? It's really just this you can think of it as like a bilinear form of this weight matrix and this correlation matrix. So a little bit of math. So we had a paper a couple years back that focused on this model where the trait of interest is a function of gene expression, some effects from gene expression, some residual SNP effects. You can think of that as a way to account for pleiotropy. I'm going to kind of ignore that for now, and some noise. And then given the TWAS, if we were to perform a TWAS one at a time, given those TWAS associations, we can define what the likelihood is. If someone were to tell us what the causal genes were, right? So if an oracle were able to say, these are the causal genes, then we can compute the likelihood of our observed TWAS statistics under this correlation framework. Obviously, the problem is that we don't know what those are, so we just place a prior on that and use Bayes' formula to get a posterior for causality. Okay, so I don't want to spend too much time focusing on the individual details of this math, but the idea is that we have a model, we're able to compute a likelihood given a causal set, and then we just flip the problem around and say, well, what's the posterior probability that any given gene is causal at a risk region? And so one nice example is that we're able to successfully prioritize sort 1, which is, has a well-known history in LDL levels. And so here what we're seeing is the predicted expression correlation matrix at this locus in, was it liver or I actually can't remember what tissue we used. And then here, what we're seeing is the TWAS association statistics. So there are several things, a bunch of genes that were null. These red lines indicate TWAS significant. So we see that there were several genes here that made it past the threshold, but what our method prioritized was a much smaller subset, and importantly captured sort 1 with the highest posterior probability. So that's all well and done. So by now what I've done is sort of introduce the concept of GWAS, how TWAS is a stopgap to understand mechanisms at risk regions. However, that comes with limitations in that at risk regions, we often see multiple hits under TWAS, and so we still require fine mapping, right? So just, and as to take a step back, the entire field of GWAS is at a crossroads, right? It's primarily been performed in individuals of European ancestry. As we see this really nice figure from Alicia Martin's paper from a few years back, that this gap has only widened as time has progressed. And then if this trend were to increase, this sort of just resulting situation could exacerbate health disparities, right? We can largely see is that, the findings that are leveraged in European populations could build better polygenic risk scores and improve care and downstream clinical practice for individuals of European descent, while those scientific gains might not necessarily translate into other populations. And so, how can we start to mitigate this issue? Well, several strides have been made in terms of increasing diversity in terms of GWAS participants. USC, I think, has been a leader in that in terms of the multi-ethnic cohort. But in addition to that, there's a large body of evidence now that says that multi-ethnic study design not only improves power in GWAS, but it enhances our biological understanding, right? And that we actually can do a better job of figuring out what's going on at risk regions when we leverage genetic data across multiple populations. And so there are two, very nice papers, another from Alicia Martin, showing how human demography affects polygenic risk prediction across populations and why, how these issues can arise. And a really nice paper from Jen Wojcik and the PAGE Consortium that really demonstrated sort of very convincingly that when you leverage large scale GWAS results across multiple populations, you do not only a much better job in your GWAS, but a better job at identifying what the causal risk variants are. And so that kind of hints at fine mapping when we talk about causal risk variants. And so actually statistical fine mapping for SNPs, right? If we want to figure out what SNP is causal, maybe not necessarily understand the causal mechanism, but just a SNP itself, kind of led the way in leveraging multi-ethnic data, for years now, starting maybe like in the early 2010s and the teens, up until more recently. So, including work here from USC, from Dr. Conti and others, that really, a multi-ethnic design is the way to go. And so given that, we sort of borrowed that line of thinking and applied it to our framework for fine mapping TWAS hits. And what we came up with was this multi-ethnic fine mapping of causal gene sets, or what we're calling it right now is just ME-focus. And so here, ME-focus leverages multiple sources of genetic heterogeneity across populations. So not only is it using differences in LD patterns across populations, but also uses differences in eQTLs, right? And so under our framework, right, our goal, again, is to identify the causal gene, not any individual causal variant at a risk region. And so based off this, the input into our method is essentially this. We require the eQTL weights for multiple populations at a risk region. We require the LD for multiple populations at any given risk region, and the corresponding TWAS Z-scores, right? And so given all of those things, we can perform fine mapping under the assumption that given a causal configuration, the likelihoods are independent across populations. So here we're using population-specific LD, we're using population-specific eQTLs, and then they just factorize given sort of, if we say what was causal. And again, we just leverage the fact that we're more interested in the inverse problem, and we just use Bayes' principle to say what's the posterior probability of any given gene explaining all of the TWAS summary statistics across these populations, right? And one of the things I do want to stress is that, again, we're able to sort of leverage the differences in LD patterns across populations, but we're not making any assumption that the underlying causal genetic variants need to be the same, right? The input to our method is just population-specific eQTLs. And so if those are completely different across populations, but the gene is still the same, our method accounts for that, whereas SNP fine mapping won't be able to. Most approaches for SNP fine mapping assume that the causal variants are indeed shared. Right, so that's all well and done. That's the model. With that defined, we went ahead and performed extensive simulations to make sure our approach works and is robust to perturbations. And so what we did is we started from real genetic data in 1,000 genomes here using individuals of European and African ancestry. We simulated GWAS data under a causal model where our assumptions are eQTLs mediate risk for GWAS. And we varied a bunch of things. We varied that eQTLs could be shared or could be independent across populations. And we compared our performance of ME focus to single population focus, just running on one population at a time, as well as a meta-analysis of the GWAS Z-scores, and then just using European eQTLs. And the reason for that is that I think common large-scale GWASs that use multi-ethnic data now just release the single meta-analyzed results, right? Where it's like across all populations, they just take the genetic data, meta-analyze those Z-scores, and it's not clear how to perform TWAS in that setting because you have a mixture of results. And so the model really is misspecified in that setting. And so that's what we compared against. And then performance also is evaluated based on the posterior inclusion probability. You can think of that as the posterior expectation for any given causal gene, as well as something we call a credible set. And the 90% credible set really is, you simply just take, if you sort the genes at a locus, just keep adding them into this set until you explain 90% of the posterior density. And this provides a way to sort of account for uncertainty at any given locus.
So Nick,
yes.
How wide of a band of cis SNPs do you consider for each gene expression model that you build?
Yeah, that's a good question. So any given locus probably has anywhere between, I want to say, like 400 to 1,200 SNPs that are common. But the number of eQTLs that then go into the model, I believe we looked at using maybe 1 or 2 in expectation and also up to like 10%.
And you're defining the regions by plus or minus 1 KB or something?
Yeah, so there are some published blocks, LD blocks, and so we just use those. So we computed what, based off those, there's this tool called LDetect, and we modified it to work in like a multi-ancestry setting. And so we have these estimates of approximately independent blocks across the genome. And then those provide the boundaries. And then given those boundaries, we just say, what's all the genetic data in 1,000 genomes? We estimate what LD is. We then, simulate continuous genotypes just under an MBN under that correlation structure to generate reference panel data for both populations and GWAS data for both populations. We then fit our predictive models in the reference data, and then perform a TWAS using those fitted weights, like under a standard pipeline.
Okay, yeah, I was going to ask about whether you had to make those selections be ethnic specific, or do you just take the superset of everything across ethnic groups?
Yeah, no, that's a great question. So I think what we did is we took them. So the algorithm works by sort of searching as you go out for, till some correlation decreases at some specified amount. And I think we just ended up saying, take the max across the populations. So we end up having like the widest window possible to account for whatever, so if LD it reaches further in one population, but not as far in the other, we still restricted a larger window.
Yeah, okay, that makes sense. Good, thanks.
Yeah. Great. And so what I'm showing here are the posterior inclusion probability, so violin plots just across all simulations for ME focus, so our method in red, versus the meta-analysis approach in green. And so down here we have the GWAS sample size. And so this should be, I think this reflects the total, either the total or like per pop sample size. But regardless, either way, we see that our approach outperforms the meta-analysis approaches across the range of sample size when they're not only when they're shared across populations, but we see like a drastic improvement when eQTLs are actually independent. And the reason for this is that you essentially just have like large model violation, right? If you just meta-analyze the SNP data under a GWAS and throw European weights at it, it's just the completely wrong thing to do. And so down here, we see similar results. If you keep GWAS sample size fixed, but vary your reference panel to predict gene expression sample size, again, we see consistent outperformance of our method versus the meta-analysis. So great, things under like, the most favorable situations, our approach works best. But not only that, do we have, higher posterior probabilities on average, looking at the credible set or the number of genes in the credible set as another metric, right? We would like to identify or prioritize fewer genes at a locus for functional follow-up. We see that again in red across varied architectures. When you change sample size in GWAS or change eQTL reference panel sample size, our approach is consistently smaller than the meta-analysis approach. And then similarly, if we look at varied trait architectures, and so here we can think of the gene effect size changing, or the variance explained by predicted expression changing. If we go from left to right in this figure, that's essentially saying, are the effect sizes getting bigger and bigger, right? And so even when effect sizes are fairly small, our approach outperforms the meta-analysis. One thing with taking a Bayesian approach is that we actually are able to say what's the posterior probability that no genes are causal at a locus. And so that's what we're seeing here. And that we can say what's the posterior probability of none of the inputted genes explaining the signal. We do a much better job of prioritizing that. And then same thing here down, here the metric just changes to the number of genes in the credible set that we're consistently seeing fewer and fewer genes in our set until you basically hit saturation with extremely large effects. All right, so again, under sort of, if we simulate under something very like close to our model, our approach beats the other approaches, so that's great, but it's also robust to population-specific architectures. And so here, if we keep the effect size in one population fixed of gene expression on disease risk or quantitative trait, but change it in the other population, our approach actually can account for that directly. And so here, what we're showing in yellow is like the single pop results in a European ancestry group. Here in blue is the African ancestry, and then again, red and green are multi-ethnic focus and meta-analysis approaches. And we see that, it's no surprise that combining data outperforms any individual population, but focusing on the red-green comparison, again, we see that our approach is much, much better in terms of assigning higher posterior probability to the causal genes. But not only that, we see improved calibration of our credible sets, right? And that as effect sizes increase, we see that our approach, if we specify 90% credible set, that we actually explain 90% of the posterior density with some standard errors. Unsurprisingly, this performance gap widens if you include additional populations. So everything we showed up until this point were just simulations based on European and African ancestry individuals. If we compare multi-ethnic focus using 2 populations versus 3, where we also include simulations for the, individuals of East Asian background, we see, again, improved performance. And so again, it's not super surprising here that it does better. It's leveraging additional heterogeneity, not only in LD, but also in eQTL diversity. Great, so what we've done so far, so how am I doing on time, actually? Well, we got plenty of time. What we've done so far is at least hopefully convince you that in simulations that not only is our approach outperforming kind of the current MO for doing TWAS and large scale data under reasonable data simulations, when we violate some of the assumptions of our model, we still outperform the others. And so next, and I want to say that the results I'm about to present here are sort of still in the works. So things are changing. So the next time I present or the next time you read the preprint, these could be totally different. Hopefully not totally, but just take these with a little grain of salt. So we applied a real data analysis on a large scale blood GWAS. So there was a recent GWAS set looked at 20 blood related phenotypes from approximately 750,000 individuals across 5 global populations. And so not only did they report, the meta-analysis across all populations, but they also report the individual population results, which allowed us to apply our method. But so great, we have population-specific GWAS and large sample sizes, but what we need also are representative eQTL data. And so to do that, we went and got gene expression from this recent study called GWAS, where they have gene expression measured in approximately 1,200 European ancestry individuals and 1,200 African ancestry individuals from LCLs. And so Zayun and Shami from my group did a lot of work in cleaning this data and getting it prepped and ready to do the analysis. Right, so imputation was performed on Genoa. QC was done over all the genetic data, sort of under reasonable assumptions, just like any other eQTL study. There were related individuals that we needed to prune. So to get unrelated individuals, that ended up resulting in 370 and 440 European and African ancestry individuals. We use the pipeline fusion to calculate weight. So that's the Sasha Gusev's TWAS pipeline. We focused on 15 blood traits, and then we performed a TWAS in each population. We performed single population focus for each population, in addition to the meta-analysis. And then we performed ME focus similarly as we did in our simulation designs. Okay, and so, just focusing on the TWAS results for a bit. So
can I stop you for a second, Nick?
Yeah, of course.
How many unique genes do you end up with in this analysis?
Oh, yeah, that's a good question. I'd have to check in. So Zane, are you on? Are you able to comment?
I think it's like 4,000 something, 4,000 to 5,000. Yeah.
Yeah. So yeah, I think so. Usually what's done to include a predictive model is whether or not it demonstrates evidence of genetic control. And so usually that's measured through heritability. And I think we use like just if expression in a given population had a heritability p-value of just a nominal p-value, think of 0.05 or 0.01, we included it. And that resulted in the current numbers we have. And I think that's pretty consistent with what other studies have seen in similar sample sizes, like if we compare to GTEx or TCGA or like.
Yeah. Okay. Thanks.
Mm-hmm. All right. So using those weights, we went and performed a population-specific TWAS. And so what I'm showing here are all the various blood phenotypes. Here are the results for the European ancestry individuals, African ancestry individuals, and then if we were to genes that were found in both. And so one thing to note here is that there's a massive discrepancy between the numbers. And so anyone, this doesn't take a keen eye to spot, but the reason for this is really differential power, right? So if I were just to hop back a few slides, we see this very impressive number of 750,000 individuals. Well, it's no surprise, based off the figure from Alicia Martin's work, that the overwhelming majority of this are European ancestry individuals. And there's a massive sample size imbalance between these. And so it's just extremely differential in terms of statistical power to identify anything. Given that we charged ahead, performed our TWAS, we see modest correlations across some of these phenotypes of our TWAS Z-scores. Nothing super impressive right now, but again, if most of the genes are null, this is not super surprising. And so here, what I'm showing are the multi-ethnic focus results. across the individual populations, European in yellow, African ancestry in blue, ME focus in red, and the meta-analysis in green. And that our approach, again, is able to output a higher mean PIP within the CS. And so, this is evidence that the credible sets output by our approach are more are higher on average, right? And that it's able to assign more evidence of causality at some given genes. One thing that we notice is that the PIPs from multi-ethnic focus do correlate very strongly with the meta-analysis, and this is really unsurprising, I think, at the moment, given how the European ancestry individuals just completely overwhelm the signal in African ancestry individuals. We are still digging through these results as we speak to try to see, what better metrics we can come up with to account for this differential power, to show that really our approach still is better highlighting genes more related to blood phenotypes. And so some of the next steps I think that we're looking at here are for the genes that multi-ethnic focus prioritizes, looking at gene set enrichment with sort of known, blood related or maybe heme related gene sets. And if we have better enrichment under our set of genes versus any of the others, that's some evidence that we're focusing on interesting biology. The other difficulty with this that I didn't highlight here is for many blood-related traits, there does seem to be quite a bit of ancestry-specific information. And so our approach does make the weaker assumption that the causal gene itself is shared. But even then, there are population-specific effects that just are observed in the GWAS that don't seem to support doing a joint analysis under a multi-ethnic focus. And so we're coming up with an additional set of heuristics based off the outputted posterior probabilities to compute some additional Bayes factors that support sharing of a gene versus population specificity. So I think those results are still a little bit underbaked, and we're not ready to share them yet. But again, this is all very preliminary. I do wish I had more real data results to convince you that this is the correct approach to do a joint analysis of TWAS across multiple populations. But in summary, multi-ethnic focus integrates GWAS summary statistics, eQTL reference panels, and LD reference panels across multiple populations. In simulations, we found that it outperforms the current practice of doing meta-analysis, and then similarly, just a single pop focus at increased sample size. And importantly, we found that it was robust to violation of some data assumptions, that it was able to correctly identify the causal gene when things varied, whereas other approaches can't. We're still trying to figure out what's going on in our TWAS results based off blood traits in European and African Americans. And we would love any sort of feedback or thoughts on how to account for this differential sample size to sort of highlight these results. Kind of that's just the nature of the field. It'd be great if we could analyze gene expression and GWAS data with matched sample sizes across populations, but just the current state of the field, that's not really, the field isn't quite there yet to be able to do that. So acknowledgements, all of this work was led by Shyamalika Gopalan. She was a postdoc in my lab for about a year, a little more than a year. She's since joined Amy Goldberg's group in Duke, furthering her education and training in population genetics. And Zane Liu, also extremely talented biostatistics graduate student who's done quite a bit of work to get things where they are. And so, this project would not be anywhere near where it is without both of their contributions, and as well as our collaborators, both here at USC and elsewhere. Whoops, I think that's it. So thanks everyone. If you have any questions, I'm happy to chat, both here and offline as well.
Nick, this is David. I'm just wondering, with the applied example, could you downsample the Europeans so they're equal, and then perform the analysis and then just see what genes do you discover as compared to sort of the ethnic specific analysis that would be discovered if you use the entire sample, or some way of kind of using that data?
Yeah, that's a great point. I think, so we only have the summary statistics, and that's one thing that we've been chatting about.
Oh yeah. Right.
So what, one maybe hack would be to sort of inflate the standard errors. If we were to estimate how sample size is affecting the standard errors in the European data, so kind of readjust them if they had been from a smaller study, and then recompute the analysis there. So I think that's certainly one approach that might be able to get at that. Some results that I didn't talk about that also try to get at this is that we assume that the effect sizes are drawn from some prior distribution and scaled by the GWAS sample size for that population. And so we do have differential kind of prior effect size or prior variances that should account for that, but it's still not quite there to address like this, just a gap this large. So we see consistent estimation of these prior effect sizes, variances across in the real data, but using them still doesn't seem to be accounting for that differential power just yet. So there's certainly a couple of other tweaks that we need to consider.
Nick, Dan here.
Hey,
Just
Dan.
wanted to ask a technical question.
Sure.
How sensitive are you to the prior distributions that you're using? How are you tuning the priors in any way here?
Oh, so I think they were fixed for the simulations. In the real data analysis, there are, so maybe I can pull up the slide if we go to the model. Where's the model? Oh, here we go. So hold on, sorry, let me share my screen. Yeah, so one thing I kind of glossed over is that we put a prior on this alpha here, so the effect of the gene integrated out, and that's why you see this variance components model here. And so this really corresponds to just the covariance across the test statistics due to just LD patterns and finite sample size, and here's the correlation across the test statistics due to whatever is actually causal at the locus. And so this is just under a normal prior. We integrate this out with some prior variance, and that prior variance is what we actually learn it in the data. So here it's this end time, this value here actually, and so we actually do like an empirical Bayes approach at each locus to learn it within each population, right? And so you get this not only the sigma squared C term, but like we know N, right? That's input from the GWAS, and so we're able to figure out what that is. The other thing that isn't mentioned here is so this prior on C. So that's just like a bunch of independent Bernoullis, because these are binary variables indicating whether or not they're causal. And so I think right now that's fixed to just like 1 in 1,000. That's something we don't try to learn from data, and we haven't tweaked at all. I think like a very reasonable change instead of 1 in 1,000 would just be like 1 over the number of genes at a locus, because we know there's signal there, right? I wouldn't encourage folks to use that in general, just across the genome. But again, we know there's evidence of GWAS signal at a region. So we know something's going on there. Something has to be explaining it, barring sort of extreme confounding or stratification or something. So given that, I think it's sensible to do something like 1 over the number of genes that essentially saying our prior expectation there is a causal gene there, right, under that probability.
Sorry, which variants are you talking about now?
Yeah, so the first thing I talked about is that we sort of learned from the data this prior variance term, and I think that seems to be kind of stable. Like learning that or keeping it at some fixed value, or like even a more naive estimator for it, doesn't change the results that much. I think what changes the results much more drastically is the prior probability for any given gene to be causal. And so the real data results that I showed. Go ahead.
No, I understand now. Yeah.
Yeah. So the real data results, I think, use like 1 in 1,000 as like a fixed probability. But I think maybe a more well, I don't know. What's reasonable to me maybe is just like 1 over the number of genes at a locus. And the motivate, like the justification for that would be that, again, we know that there's evidence, like strong evidence of genetics influencing risk or some quantitative trait. Right? These are done at GWAS risk loci. They're not done just blindly across the genome. So given that, right, we know there's at least one gene. And so if we just do it 1 over the number of genes, you know, that's just saying an expectation there's a causal gene there. But, you know, certainly that's, you know, that's under still some assumptions that it's a gene and not any individual isoform or splice junction or whatever. So, but it might be, you know, a stronger starting point that boosts power. So
You can't do cross-validation to get at that.
If we have a set of genes that where we know they're causal, we could, you know, so certainly if we change maybe our phenotypes that we're looking at, or, you know, dug deep into the literature for these blood phenotypes, if folks have some prior validation, like, you know, genes that have been functionally validated that actually influence the structure of like these blood traits, or like, you know, the phenotype, whatever these measurements are, right? Like the mean cellular hemoglobin levels or like platelet count, stuff like that. I'm not sure if that's out there though. That would be one way that we could, but certainly that would be great if we could include that in the manuscript. It's a good idea.
But it's not something that you can learn from your data.
I mean, I can see maybe taking an empirical Bayes approach to learn that, but I don't know if learning that at the same time with that prior variance complicates anything. But that's something that we could certainly check.
And there's a large literature in Bayes model averaging and model selection for the prior on that parameter beta binomials and how that influences the model selection and so on. So.
Oh, cool. Okay.
You know, the variance, though, it's hard to estimate.
Yeah, I'll definitely, I'll have to give, maybe take a deeper dive into that to see what's been done. Because my only concern is that, you know, trying to not only leverage, you know, or learn the prior probability, but if the variance, you know, I feel like there's some multiplicative effect there that you could like push weight from one to the other, that maybe it's not identifiable if you want to do both at the same time. But you know, this is all like my intuition. That's not any sort of proof of anything.
Hi, Nick. This is Linda.
Hey,
Can
Linda.
you hear? Hi. I joined the call late because I just saw your email later. So I missed which data set you were working with. But have you thought about going to like TopMed or other consortia and focusing maybe on a region that is known to be highly correlated? Yeah.
Well, I would love to do that. Absolutely. I think, you know, we need to get this paper out there first, to sort of demonstrate to the you know, the broader field that like this tool does what it should, and you know, at least with some preliminary results, that, you know, find sensible things, and you know, folks should trust the results. But yeah, I mean, I would be thrilled to have it applied to TopMed if that was a possibility.
Like, I'm not sure if it's feasible, but just thinking off the top of my head, like if they've called, you know, in a very detailed manner, say the HLA region, and then have more confidence about that region, like to tease out those genes kind of by ethnic group.
So the HLA region is like the one region I would say for sure don't run this tool.
Okay.
Unfortunately. And the reason is just because LD is just way too complicated there. And I don't know if leveraging multiple populations, like heterogeneity in LD across populations, is going to be enough to figure out what's going on there. I could be wrong, right? And, you know, and in fact, actually, I'm not sure if this is some folks have investigated this in depth at that locus. I know for sure at a single pop level, like I would definitely just drop any sort of results that were run in that region because of the pervasive LD. But you know, that is an interesting question. I'm not sure actually if folks have looked at multi-ethnic analysis, and you know, I'm sure they have, and this is just me being ignorant of like maybe what's been done in the field. But
yeah, It might be worth checking into, just because I know, like at the DCC, I think it's Marcia Wheeler has done detailed calling, which might have helped, but I'm sure there's other regions too, besides there, that might be, that you could compare it to or something.
Yeah, cool. I mean, we should definitely chat more offline to see, to figure out some possibilities there. I would love for folks to get this in their pipeline.
Hi, Nick. This is Mateo Cesar. Thanks for the talk.
Hey, Mateo, how's it going? Thank
You mentioned at some point that you don't assume the genetic architecture to be the same across populations. So does that mean that the causal genes may not be the same?
Well, so yeah, that's a good question. So so right now our assumption, when I say genetic architecture, I guess I'm more, it's a looser, not quite as strict a definition as that. What I'm referring to is that the causal gene is the same, but the mechanism, like the SNPs that regulate that gene, are allowed to differ across populations. And really, it's because we're not making any assumptions. Like they could be the same, right? But we're not making any assumption on what the regulatory SNPs are. And that's purely guided by the eQTLs learned within each population-specific reference panel. And so here, right, we just take GWAS, the signals are whatever they are in each population. We take their LD and we take the eQTLs for the same tissue, but in each population, and leverage those effects. And that way, if you do have sharing of eQTLs, that if the same SNP regulates the expression of that gene across both populations, that should be reflected in the eQTL data. If they are different, that should also still be reflected in the population-specific eQTL data. And so our model's kind of agnostic, and it just says, feed it in the eQTL data, and whatever the architecture is, it is. But it does assume that the causal gene is shared, and that there are no interactions or anything like that. Those, under strict assumptions of linearity and all that.
Thanks.
Well, cool. Any other questions? Hey, Stephen.
Yeah, hey. Yes, I have one. Yeah, so I'm wondering if you can leverage Biobank Japan, where you have large summary statistic in East Asian. But I don't know what is the biggest sample size you can get in terms of gene expression for East Asian or Japanese populations.
That's the problem. That's entirely the problem, is that the things like Riken, or Biobank Japan have massive sample sizes in terms of GWAS, right? That would rival things like UK Biobank and would certainly solve the issue of differential sample size in the GWAS level. I think the primary issue is that just the eQTL reference panels don't exist.
Okay.
And so
is that they not exist, or they are not shared with the community?
They're not shared. They're not shared.
Okay.
I think they do exist. I think there are I think Riken does have RNA-seq data, but I don't think they've shared they've done the analysis yet and shared it with the community. So so if the weights were out there, if eQTL data were out there, then certainly we would just hop right on that and at least get rid of this issue of differential sample size.
Yeah. Okay, I think maybe not for this paper, but I think for another project, I think it could be interesting to reaching them. I don't know, my sense is that they have a lot of data, but they don't have a strong StatGen community, developing methods. And I think I'm pretty sure they will be interested in applying your tool.
Oh, I hope so. Fingers crossed. I want everyone to apply it. Everyone to. So awesome. This is all a lot of great questions and feedback. I really do appreciate it, and I appreciate being able to present to everyone today. And so if anyone has any other questions, certainly contact me or Zane or Shami. We're all really happy to talk. And if folks have thoughts on things we can include, certainly the manuscript isn't written yet, so we're open and open arms, willing, open papers. I don't know, whatever you want to call it. Happy to chat more. So. Cool.
All right. Well, thanks, Nick. That was a great talk. And thanks, everybody, for attending.
Great. Thanks.
Thanks. It was terrific.
Yep.
Take care.
Okay, you too.